Meiosis. Okay, so we're going to do our diagram. We're basing this off of textbook pages 196 and 197, and I had you in the video yesterday draw in interphase, hopefully to keep you from messing up when you're doing your diagram here. You do need 4 different colors. You don't have to use the same colors that I'm using, but I happen to have a red, blue, green, and yellow, so that's what I'm going to be using. You're going to start off with just a resting cell. So we've got a nucleus, and we have just our unraveled DNA in our nucleus during interphase. And so then we're going to go to prophase 1. In prophase 1, our nuclear membrane begins to break down, so we draw a dotted line, and our chromatin condenses into chromosomes, and so they take on this X shape, and we end up with these, okay? And each of these chromosomes has a little center, I kind of call it a button, but it's called a centromere, and it more or less holds those 2 chromatids together. When we get to metaphase 1 in meiosis 1, the chromosomes still line up in the middle, but this time they are actually in pairs. So let's say the red and the green are a pair, and the blue and the yellow are a pair, and so they line up in pairs. And there's still the spindles that are going to eventually pull them apart, and that is metaphase 1. And so when we get to anaphase 1, we have whole chromosomes that are on each side of our cell, and each side still has a full set of chromosomes, but they're not identical. They're very much different. And so this is different from mitosis. Remember, in mitosis, they lined up in the center, all single file, and half of each one ended up going to each side. Whereas here we have the whole chromosome going, but they still have the same number. So when we get to telophase, we have our nuclear membrane forming in each of our daughter cells, and here we have the red and the blue chromosome in that one, and we have the green and the yellow in this one. Now, I'm going to add something to these so that we can kind of trace. We said that homologous chromosome pairs have the same size and structure, but they carry different versions of the same gene. So let's say that the red and the green chromosomes have a gene for eye color. And let's say that on our this part of the chromosome, we're going to put a mark here, and that's going to mean that that's for brown eyes. And if it doesn't have the mark, that's going to be for blue eyes. And then on our red chromosome, we're going to have a mark maybe over here, and that's going to be for green eyes. And if it doesn't have it, then maybe it's brown eyes. Okay? And so we're going to do the same thing. Let's say on the blue and the yellow, there's a trait or a gene that is going to be hair color. And so we're going to have this will be blonde hair, and if it doesn't have it, it'll be brown hair. And then on the blue one, we're going to have red hair. We're going to put that one over here, okay? And so that'll kind of help us watch that these are going to end up being different genetically when we finish, okay? So once we go through telophase, we are not going to have interphase where the DNA duplicates. So we're starting out essentially in prophase. The chromosomes are already condensed, and so we might even say that, I could have done that a dotted line, I guess, to show that that nuclear membrane is starting to break down. And each of these daughter cells has a full set of chromosomes, but they're not the same. And so we end up copying pretty much what we had there in telophase. So we've got that. Now each of these daughter cells is going to separately go through metaphase, where the chromosomes again line up in the middle. And so I'll put one here, and I'm going to add, remember this is a gene for eye color and hair color, and then over here I have them lining up in the middle. And don't forget to put the gene for eye color and hair color. And I'm going to add my spindles in here that are going to attach to that centromere. Put that little button in the middle. I forgot to put that up here. Okay, so they're going to attach to that. And so when I get to anaphase of mitosis 2, or anaphase 2, now my chromosome is actually going to be split. So in anaphase 2, I have the spindle fibers, and I have these being pulled apart, and I'm going to again make sure I have my different genes shown here. Okay, so now you can start to see that, of course, these two are not identical. We know that they're different colors, but even these two cells are going to end up not being totally identical, because this one has traits that this one does not. It has features that that one does not. So we're going to go ahead to telophase, and we're going to draw our nuclear membrane dotted lines so that we know the nucleus is forming, and it's going to eventually go through cytokinesis and have different cells, separate cells, and we're going to see those features in this last row. So in telophase 2, we notice that these two cells are not even identical to each other, because this one has these traits, this one does not. Same thing goes for here. So when we end up with 4 haploid cells, and these haploid cells have similar DNA, but not identical. So we have 4 non-identical cells, and they have half of the genetic material. And so when they combine with another sex cell that has half of the genetic material, they will actually have the total chromosomes of that organism. So on the back of your meiosis notes, it goes through meiosis 2, which is prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. And then it says, how how does meiosis compare to mitosis? So compare means tell me how they're alike, tell me how they're different. So both mitosis and meiosis start with a full set of chromosomes. So both of them start with a full set of chromosomes. So that'd be 23 pairs or 46 chromosomes for humans. They are both a form of cell division. There's my little shorthand, division. Both a form of cell division. So mitosis results in 2 diploid or identical cells that have 46 chromosomes, and meiosis results in 4 haploid non-identical cells that have 23 chromosomes each. The cells that go through each of the processes are different. Body cells go through mitosis. Sex cells, the sperm and the egg, go through meiosis. During mitosis, the nucleus divides 1 time. During meiosis, the nucleus divides 2 times. And finally, the last difference, mitosis only has 4 steps, PMAT, and meiosis has 8 steps because it has all PMAT1 and PMAT2.